Let's look at some of the sources of nitrogen. Where do you get nitrogen from? How does it come in? There are natural sources of nitrogen. We have what we call symbiotic nitrogen fixation. Symbiotic nitrogen fixation occurs when you have bacteria of the form that we call rhizobia. These are associated with plants in the Fabaceae, the bean family. The combination of the two causes a symbiosis that allows for the plant to provide the bacterium with sugars and allows the bacterium to provide the plant with nitrogen. These are done by bacteria in the genus Rhizobium. There are a lot of different rhizobia depending on the species of bean plant. We also have non-symbiotic nitrogen fixation. Non-symbiotic nitrogen fixation is where you will have bacteria that are associated with the root zone or free living in the soil. These will fix their own amounts of nitrogen. Then as they degrade, this will supply nitrogen to the plant. These are prokaryotic organisms that do this. We're talking about free-living soil bacteria and free-living blue-green algae. One of the genera that we talk about is Frankia, which is an actinomycete. When we're looking at symbiotic nitrogen fixation, we have a process by which the nitrogen is fixed. There is a protein called leg hemoglobin. It is red in color. It is hemoglobin-like, hence the name leg for legume, hemoglobin. Basically, it allows for pickup of nitrogen and conversion of the nitrogen into ammonia. Non-fixing nitrogen nodules on plants are green. When they are fixing, they are red in color due to the leg hemoglobin. The reaction where you have nitrogen gas plus 8 electrons plus 16 magnesium ATP plus 16 waters, providing you with 2 ammonia plus hydrogen gas plus 16 magnesium ADP plus 16 organic phosphorus plus 8 hydrogens plus 16 hydroxy units. You do have the ammonia out of that. That can be used by the plant. Here's your process. What happens is you've got root hairs on the plant. The root hairs come in contact with rhizobia bacteria. The rhizobia bacteria cause a curling of the root hair, and on the inside you get what is called an infection thread. This is really an invagination of the cellular membrane, which is going to allow the bacteria to migrate down through the center of it. The Golgi vessel down there produces a lot more membrane. That allows your infection thread to extend. It eventually extends down into the intracellular space below that epidermal cell that produces the root hair. Then you're going to get colonization down in this bottom part with these infection threads which go down into neighboring cells. The presence of the rhizobium starts gall formation on the root. These little galls will then become your nitrogen fixing nodule. All species of rhizobia have the ability to produce these increased sizes and growth, which are really kind of a nodule. Those nodules therefore become very important. This is a picture of nitrogen fixing nodules on a plant. You can see in this particular case, rather than being round, they're kind of elongate and flattened. This depends on the bacterium, depends on the plant tissue itself. This is the comparison between chlorophyll on the left-hand side and leg hemoglobin in the center. You actually see a hemimolecule. You can see nitrogen is contained in both of these. In the chlorophyll, you've got a magnesium in the center, which gives chlorophyll its green color, whereas in the hemi-type molecule, the hemoglobin, you've got an iron on the inside, and the iron provides the red color to this. When we look at nitrogen fixation, it is producing ammonia. The ammonia goes into amino acids or into nitrogen-rich compounds. Some of these nitrogen-rich compounds can be stored. The amino acids can be migrated around inside of the xylem to other places where they might be needed. We also have the formation of urides. Urides are one of the materials that are considered to be these nitrogen-rich compounds. Urides would look like this. They include allantoin, allantoic acid, and asparagine. Asparagine is a type of amino acid. When we look at these, you can see that they've got high levels of ammonia relative to the amount of carbon. Then these can be transmitted around and they can go to various places within the plant body.